So just to give a bit of context, Scots pine and Dothystroma needle blight, this is the species and the disease that I'm going to be talking about. So Scots pine has got this huge distribution right from the Atlantic to the Pacific and right from the Arctic Circle down to the Mediterranean. And the distribution that we've got in the UK, the sort of native distribution, is its westernmost part of the range. And the fact that it grows in such a vast area means that it's very, very phenotypically diverse. And globally, the most important disease of pine is Dothystroma needle blight, which is caused by a pathogen called Dothystroma septosporum. And it causes these very distinctive lesions on the needles. And when you get a lot of disease, as you can see in this middle picture here, it causes needle loss and growth retardation. And it can, in severe cases, also cause mortality. But the main reason that people are concerned is just really this sort of growth, the growth effect that it has as well. And so I'm sure it doesn't need pointing out to this audience, but understanding and predicting host susceptibility is crucially important to effectively manage both existing forests. In this case, I mean both the sort of native standing populations, but also plantations as well, but also for planning the new forests that we're going to be planting. And so a bit of background for Scots pine and forestry. It's really, really handy actually having the background for Sitka already, because Sitka is by far and away the most well represented in terms of forestry. You can see that in terms of the stocked area and also the seedlings, Sitka sort of dominates, but Scots pine is the second most important species in the UK. There was a breeding program that sort of kicked off in the middle of the last century and involved domestic plantations and native pine woods. And there were selections and progeny test series. And then there was a reselection of parents from the general breeding population selected for height. Oh, sorry, I'm going a bit too fast, apparently. Apologies for that. Selected for height and straightness in 2002, and after this the program was closed. So but having said this, 50% of seedlings sold are still from seed orchards, so they're still a really, really important source of planting material. And you can see that in the last few years there's been an increase in plantings or in seedling production from Scots pine. And in order to understand why it is that Sitka is so preferentially chosen over Scots pine, you know, what drives these choices, I was involved in a project a few years ago that Richard was also in, and their part of the project was looking at what the stakeholder priority traits are in the UK and what drives decision making with regards to species choice for planting. And somewhat surprisingly, the biggest priority for both, well, overall for public and private stakeholders was disease tolerance. It kind of came well above climate resilience, but also, interestingly, tree form. In private stakeholders, tree form was more important, but overall disease tolerance. And there was seemed to be this need to have confidence that what was being planted would be tolerant to disease in the same way that people can rely on Sitka at the moment to be relatively robust against the threats. So as a result of this, this is the work that Richard's been leading in this project. We're aiming to sort of restart the Scots pine breeding program and build up those genetic resources and try to infuse what we understand about Dothystroma resilience or tolerance into this breeding population. And so this part of the work has involved taking two distinct sources for the genetic resources. So one is a native pinewood progeny provenance trial that we have, which I'll talk just about in a second. And these have been characterized for susceptibility, but also for growth and straightness and branching as well. And there are also the existing clone banks and seed orchards as well. And so from these genetic resources, the project has been working on performing controlled pollinations and also taking grafts in order to then have progeny trials ready to go, and also to develop a breeding arboretum with which to archive this material. So this is a really important strand, so very practical, that's being done and being led by Richard. And I mentioned the progeny provenance trials. So just a little bit of background about those. So this is a set of 3 trials that we have planted across Scotland. So there's one in the northwest of Scotland, one in the northeast, and then one down in the borders. And these are comprised of trees originating from 21 native pine woods. And they have this family structure as well. So there are 8 families per population. And they were established in 2007. The trees were planted out in 2012, and they've been regularly assessed for growth, for phenology, for susceptibility, and also for reproduction. You can see here, this was work that was done a few years ago on the bottom left. And some work was done looking to understand variation in susceptibility to Dothostroma, so DMB, Dothostroma needle blight, but also height, and working out what sort of gains could be made by reselecting particular families. So sort of coming back to the real fundamentals then, I've said that these trials have been assessed for susceptibility. And this is relatively straightforward to do when you're able to go back to the same trees time and time again. You've got the time to do that. Things are laid out in a nice sort of common garden, progeny provenance setup. But in order to assess variation susceptibility, there's actually a few different ways that we can do this. So you can do an artificial inoculation, and this is inoculating plants in a controlled environment with selected pathotypes. And you do this for a defined period, and then you would then analyze everything. A natural inoculation is what I mentioned before, so that's what we've done with this progeny provenance trial. So this is planting a common garden trial, so everything's exposed to more or less the same environment, and you know that there's going to be infection in the area, and then you track the progress. I should say, actually, the trial that I'm referring to, we didn't plant it thinking it was going to get infected, but by this point, Dothostroma seemed to be more or less everywhere, and so once we realised there was infection in the trial, we started assessing it. Another approach to assessing variation in susceptibility is just to survey what's there. So you can go out to forests, you can assess the standing trees, and you just record it. And then finally, there's the option of a detached needle bioassay. So this is where you inoculate individual needles and you maintain them in a controlled environment for a defined period of time. And you can see I've put ticks against the first 3 because those are ones I've been involved directly with. The first 2, and forest surveys are fairly routinely done. But detached needle bioassays are not something that we've done before as an approach to understand susceptibility to Dothostroma. So we wanted to really see if we could develop this as a tool. So when comparing the different methods, you can see here that there's sorry, it's not done the animation as I wanted it to, but anyway, I think it's fine. You can see that if we just kind of simplify everything, there's a lot of differences in terms of the experimental design and in terms of the practicalities here. But if we just take 2 of them, we've got host variation. You can control the host variation that you're analysing in artificial and in a natural inoculations. You really can't in a forest survey. It's just whatever happens to be planted there. But in detached needle bioassays, again, you can kind of select which of the trees are going to be included. And when it comes to practicalities, a really important point is the complexity. And with complexity, I'm kind of combining lots of things. So I'm talking about how expensive it is, how much time it takes, how much space you need. And artificial inoculations and natural inoculations both require a lot of time, resources, and they tend to be fairly expensive because you need to have both of those in ready supply. In comparison, forest surveys are relatively inexpensive and simple. You just turn up and you do your assessment. And similarly, detached needle bioassays, they're relatively simple. You have to have a bit of space for them, but you just need some bench space. You don't need to plant them out in it and let things grow and grow. So some of the questions that we had coming into this then were, how well do bioassays reflect susceptibility in the field? Also, can bioassays be used to identify susceptible and resistant trees? And also, how consistent are the results from the same tree or from the same clone, if we're talking about grafts, sampled at different spatial scales, so that we can get a feeling for actually how useful the method is? So yeah, my research question. So first of all, then I was asking how well do bioassays reflect susceptibility in the field? And so for this, I used this Common Garden progeny provenance trial, which has been surveyed for susceptibility from, well, so near enough 10 years now. And I'm not going to go into detail here, although I'm very happy to explain about it. But each year, with a couple of gaps, I've been assessing every single tree in the trial. And we've got a nice distribution of trees ranging from sort of high to low susceptibility. And for those, I've got a sort of a individual susceptibility value for each tree. And I've also assigned them based on the sort of tail ends of the distribution. Some are assigned as resistant and some are assigned as susceptible based on their values. And so these trees were then used as the source for bioassays. And the sort of hoped outcome is some sort of validation. So bioassay susceptibility should correlate nicely with field susceptibility. That's the hope. So my second question was, how good are bioassays at identifying susceptible and resistant trees? Now, in this case, I realized that it's susceptible and resistant is can be quite subjective. It can be related to the environment. So like I say, I've defined in this environment which trees are susceptible, which are resistant, and I want to see if the bioassay picks up those same patterns. So the same trial again, put into bioassays, and in this case, the susceptibility in the bioassay, you look at the distribution, you assign some of these as resistant, some susceptible. We've got a cutoff of about sort of 15% tail ends of the distribution to try and kind of capture just the extremes of the trait variation. And then assigning these groups, how well does these groups then correlate with field susceptibility, and also can trees which are identified as resistant in the bioassay correspond with those which are resistant in the field as well. And hopefully we've got a sort of accuracy outcome from this. And finally, I would like to know how consistent results are from the same tree sampled at different spatial scales. So for this, I considered sampling within a single tree, so within different positions in the crown. Sort of a moderate spatial scale, so within a single site, but ramets. So ramets are the same genotype, the same individual tree, which is then grafted onto different rootstock and then planted and sort of grows into different trees. So ramets should be genetically identical individuals. So when you've got multiple ramets in the same site, how do they look in the bioassay? And also large scale, so the same clones that are planted at different orchards. So these final two, the sort of moderate and large scale, these come from trees which are planted in seed orchards, whereas this small scale comes from the Common Garden trial that I mentioned before. And these are all popped into a bioassay. And then again, I want to look and be able to compare results for this to be able to assess some sort of reliability score as well. So I've been talking a lot about the detached needle bioassay, but what do I actually mean by this? So I've tried quite a few different approaches, and this is what I've ended up with. So you start by collecting your sample from a tree, you store it in just a poly bag and keep it at ambient temperature, and then when you get back to the site, you pop it in the fridge, keep it chilled. And then when you're ready, you take individual needles from each branch and you pop them in a specified location. And you repeat this. So this is one block and this contains one needle pair from every single tree. And you might repeat this 10 times so that you've got 10 replicates of the same setup, just randomized each time. And keeping the needles wet, because really we want to make sure that we're especially in the very beginning stage, we wanted to make sure that it was sort of replicating a sort of, what might happen if you're exposed to or susceptible to Dothostroma, and that generally comes along with having a lot of needle wetness, because that's conducive to pathogen growth, dispersal and development as well. And then what I was doing was assessing the brownness. So brownness is very, very simply just what percentage of each needle pair is brown at each point. So this is 21 days, after the start of the experiment. And then again, I did another measure at 49 days after the experiment, so about 7 weeks. And then quite simply, that was it. I just look at brownness as it progresses over the course of the period of about 7 weeks. And then there's take the data and then perform these steps that I was talking about before. So validation and accuracy, and actually see what we can get from it. So you might be wondering why I've not mentioned at all about inoculating the needles with Dothystroma spores. Why wouldn't I do this? Well, I did. I've done a lot of inoculating detached needle bioassays with Dothystroma spores, and I was constantly coming up with issues because of pre-infection. So Dothystroma is more or less everywhere in the landscape. And I tried many different things to try and sort of prevent or minimize the risk of pre-infection. I sampled at different points in the year. We even tried isolating the needles in the bags that are used for the controlled pollinations to try and stop the spores from coming in, and it was just, yeah, it was just not working. And it means it's very hard to be able to disentangle the results from pre-infection with those from inoculation. So I also found needle brownness, to be honest, was a sort of I measured it at first just because I noticed that some were turning very brown and some just do not turn brown at all. They can be in a wet box for 7 weeks and they remain totally green by the end. And there were other added benefits as well. It's much quicker. Just the scoring is very simple. It's cheaper. You don't have to then start buying consumables and reagents to do an inoculation, and it's safer. I wasn't chopping, cultures out with scalpels and things like that. So overall, I was thrilled that needle brownness was, a much more effective way of trying to understand susceptibility in a bioassay environment than inoculating. It was a good result. So for my first question, I was asking, how well do bioassays reflect susceptibility in the field? So just for a little bit of context, this is, if you look over a year, then where I've put orange, this is where there's really not much in the way of growth. You might get a little bit at the tail ends here, depending on how warm it is. But generally speaking, bud burst progresses from about April, and you get, it varies quite a lot, but you get it continuing. So you get the full needle emergence and elongation by about June. And then you get active growth happening over this period, and then it slows down again by the time you get to sort of October time, again, slightly depending on the weather, on the climate. And so I did bioassays where I collected needles in July, in August, in October. As I said, at first I was trying really to minimize the chance of pre-infection. So I thought that sampling earlier would actually be much better. But you can see there's really no relationship at all if you look at bioassay susceptibility compared to field susceptibility. And in fact, you can see that bioassay susceptibility, most of the trees were really highly susceptible. And this is because the needles were so brand new, newly emerged, they just succumbed to the bioassay conditions really quickly and all turned brown very early. In August, we see a slightly better pattern, sort of a tentative positive association, but a huge amount of noise. But by August, we see actually quite a nice, clear, again, there's still quite a lot of noise, but that's fairly standard. But we at least see a really highly significant positive correlation between bioassay susceptibility and field susceptibility. So I was really pleased with that. Validation, tick. So my next question was, how good are bioassays at identifying susceptible and resistant trees? And similarly, I just used the same approach over these 3 time points. And you can see that actually in the bioassay, trees which were assigned as susceptible do have overall higher field susceptibility, but the difference isn't significant until we get to October, and then you see really highly significant differences. So trees which are assessed as classified as susceptible based on their bioassay do correspond with significantly higher field susceptibility values as well. And you can also see that the accuracy, which, I mean, we can pretty much ignore July and August, seeing as they're not useful. You can see that the accuracy is actually higher for susceptible trees. And you can see that here, the box plot here is much smaller. It seems to be much more accurate to define susceptible trees compared to resistant. And the error rate is much lower as well for susceptible trees. So it might mean that this method is actually particularly useful for identifying susceptible trees. And so my next question was, how consistent are results from the same tree sampled at different spatial scales? So first of all, there was a small scale. So this is branches sampled from high up in the canopy, in the middle of the canopy, and low in the canopy. And I should say these trees are now about 17 or 18 years old. They're generally sort of between about 4 and 6 meters. And you can see that there is no significant difference among any of these, whether you sample in bioassay susceptibility, regardless of where you sample them. And you can see that they're very highly correlated as well. Susceptibility is for trees, for needles sampled in the bottom of the crown, is highly correlated with susceptibility for needles sampled in the middle of the crown. Slightly less between the bottom and the top. But what you find is if you then just look at those trees which are assigned as resistant or susceptible based on their field susceptibility, you see actually there is only a significant difference if you look at needles which are sampled in the middle of the canopy. And I think possibly the reason for this is that because these trees are starting to drop their lower branches, I think there might be something to do with needle condition in these bottom branches, which just makes them less reliable. Similarly, in the top of the canopy, you've got exposure to a whole range of more extreme environments in terms of the abiotic and biotic environment, or particularly the abiotic environment. So again, it might be that crown position is associated with needle condition, and in the more benign environment in the middle of the canopy, that's where it becomes easier to pick apart those differences. It's possible that you see a reduction in this effect once canopies close, but that's something I need to look into. So there's sort of a tick for reliability, but, you know, I think a little bit of cautiousness there. And then I looked at moderate and large scale spatial scales. And so you can see that for the moderate spatial scales, so this is looking at the difference among ramets of the same clone, you can see that there isn't a significant difference. So it suggests that using a single ramet per site may actually represent a clone susceptibility at that site. The caveat being that orchards show highly significant differences. So you can see here, these are 3 of the orchards. And although, you know, quite often the trend is in the same direction, I couldn't do a proper genotype by environment interaction here because not all of the genotypes were present in all of the environments. But you can see that there are some interactions, but generally speaking, things move in the same direction. But clearly there is a big effect of site here as well. And it's probably just the case that trees are growing for a really long period in a particular environment, and that has big effects on the needles as well. So I would say apply caution to the use of trees from different environments. So there's sort of a plus point, you know, it's really good that you can use, you know, within sort of moderate spatial scales, there isn't a big effect, but I think just be very careful about inferring things from trees grown in different environments. So some of the recommendations based on this work, I should say this is just like a little part of the work. I tested it in other situations as well. But the recommendations sort of for the basics are to collect later in the growing season if possible. We think this has something to do with sort of robustness of the needles. Avoid directly comparing susceptibility of trees which have been grown in different environments, as I said. Apply a consistent sampling methodology. So make sure you're sampling from the same location within the crown. Include as many trees as possible in each bioassay. I thought at first if you just, you know, I could just include a very small number, but actually you need that a scale in order to be able to pull apart those tail ends. I just think it's not so useful as a technique otherwise. And be really cautious, much more cautious when classifying trees as resistant as compared to susceptible. So the next steps, I'm going to repeat the bioassays using the same trees and the same time of year, just to make sure that we've got some measure of reproducibility. I'm also interested in using younger seedlings because it's possible there's an ontogenetic response, which means that it's a response based on the age or its stage of development. And really for this approach to be useful, we also want to be able to use it to assess susceptibility in very young seedlings. I'm really interested in comparing artificial inoculations alongside the bioassays to compare the response, and also to understand the mechanisms underlying this response. And a little bit more about this, I think what we're potentially seeing here, one of the hypotheses is that the response we're seeing isn't a sort of a direct, it's not directly saying that this exact same mechanism is at play. It's possible that the bioassay is picking up, it's a sort of a proxy for susceptibility because there might be similar sort of needle structure. There is something to do with the needles which responds in a similar way to the conditions of the bioassay and kind of is able to slow down that browning response, which is also something that we see in the response of susceptibility. So I'm really keen to understand, and actually we've got a phase 2 CFP project, and part of this is going to be doing a little bit more work on looking at the sort of mechanistic processes underlying this susceptibility response as well. And then also what I'm keen to understand is what's the optimal timing for this? So as I said, Brown has started off as a bit of a, oh, that's interesting. So I hadn't really thought about the best timing to do. And so I think if I can conduct weekly surveys, I could then optimize this process further. So just to say that there's a lot of people involved in various aspects of this work. The lists here, massive thanks obviously to Richard as well. Yeah, thank you all for listening.